Biosensors. A biosensor is an analytical device which employs a biological material to specifically interact with an analyte. This interaction provides some detectable physical changes which is measured and converted into an electrical signal by a transducer. Finally, the electrical signal is applied, interpreted, and displayed as analyte concentration in the solution preparation. An analyte is a compound whose concentration is to be determined. In this case, antibodies, lectins, whole cells, entire organs or tissue slices are also being used. The nature of interaction between the analyte and the biological material used in the biosensor may be of two types. One, the analyte may be converted into a new chemical molecules. By example, such biosensor are called a catalytic biosensor. And two, the analyte may simply bind to the biological material present on the biosensor. Example, to antibodies, nucleic acid. These biosensors are known as a affinity biosensor. A successful biosensor may have at least some of the following feature. 1. It should be highly specific for analyte. 2. The reaction should be as independent of factors like stirring, pH, temperature, etc. as is manageable. 3. The response should be linear over a useful range of analyte concentration. 4. The device should be tiny and biocompatible in case it is to be used for analysis within the body. 5. The device should be cheap, small and easy to use. 6. It should be durable, that is, should be capable of repeated use. A biosensor has two distinct type components. 1. Biological, example enzyme, antibody, etc. 2. Physical, example transducer, amplifier. The biological component of biosensor performs two key functions. 1. It specifically recognizes the analyte. 2. It interacts with it in such a manner which produces some physical changes detectable by the transducer. These properties of a biological component imports on the biosensor its specificity, sensitivity, and the ability to detect and measure the analyte. The biological component is suitably immobilized on the transducer. The enzymes are usually immobilized by glutaraldehyde on a porous sheet like lens, tissue, paper, or nylon net fabric. The enzyme membrane so produced is affixed to the transducer. Generally, correct immobilization of enzyme enhances their capability, which may be rather dramatic in some cases. As a result, many enzyme immobilized systems can be used more than 10,000 times over a period of several months. The biological component interacts specifically to the analyte, which produces a physical changes close to the transducer surface. This property changes may be 1. Heat released or absorbed by the reaction measured by the calorimetric biosensor. 2. Production of electrical potential due to the change distribution of electrons, potentiometric biosensors. 3. Movement of electrons due to redox reactions, amperometric biosensors. 4. Light produced or observed during the reactions, optical biosensors. Or 5. Change in mass of biological component as a result of reactions, acoustic wave biosensors. The transducer detects and measures these changes and converts it into the electrical signal. This signal is necessarily very small, and it is amplified by an amplifier before it is fed into the microprocessor. The signal is then proceeded and interpreted, and it is displayed in suitable units. Thus, biosensor converts a chemical information flow into an electrical information flow, which involves the following step. 1. The analyte diffuses from the solution to the surface of the biosensor. The analyte reacts specifically and efficiently with the biological component of the biosensor. 3. This reaction changes the physiochemical properties of transducer surface. And 4. This leads to a change in the optical or electronic properties of transducer surface. 5. The change in optical electronical properties is measured, converted into the electrical signal, which is amplified, proceed and display. Type of biosensor. 1. Calorimetric biosensor. Many enzymes catalyze reaction produces heat exothermic. The calorimetric biosensor measures the change in temperature of solution containing the analyte following enzyme action and interpret in terms of analyte concentration in the solution. The analyte solution is passed through a small capacity 1 ml pack bed column containing immobilized enzyme. The temperature of the solution is determined just before entry of the solution into the column and just as it is leaving the column using the separate thermistor. This is the most generally applicable type of biosensor, and it can be used for turbid and strong colored solution. The greatest disadvantages is to maintain the temperature of the sample stream at a constant, say plus or minus 0.01 degree centigrade temperature. The sensitivity of 10 to the power of minus 4 molar and the range of 10 to the power minus 4 to 10 to the power minus 2 molar of such biosensor is quite low for most applications. The sensitivity can be measured by using 2 or more enzymes of pathway in the biosensor to link several reactions to increase the heat output. Alternatively, the multifunctional enzymes may be used. Example, glucose oxidase is used for determination of glucose. 2. Thermistor containing biosensor. The thermistor is used to record even a small temperature changes between 0.1 to 0.001 degree centigrade during the biochemical reactions by immobilizing enzymes like cholesterol oxidase, glucose oxidase, invertase, tyrosinase, etc. The thermistor have been developed. Moreover, thermistor are also employed for the study of antigen antibody with very high sensitivity 10 to 13 molar dm-3 in cathode used in case of thermometric enzyme linked immunosorbent assay ELISA. 3. Potentiometric biosensor. These biosensors use ion selective electrodes to convert the biological reactions into the electronic signal. The electrode employed are most commonly pH meter glass electrode for cations, glass pH electrode coated with a gas selective membrane for carbon dioxide, ammonia and hydrogen sulfide, or solid state electrode. Many reactions generates or use up H plus, which is detected and measured by the biosensor. In such cases, very weakly buffered solutions are used. The gas sensing electrodes detects and measures the amount of the gas produced. An examples of such an electrode is based on urease, which catalyzes the following reactions: CONH2 twice plus 2H2O plus H plus gives us a 2NH4 plus HCO3. This reaction can be measured by a pH sensitive ammonium ion sensitivity, NH3 sensitive or CO2 sensitive electrode. Biosensors can now be prepared by placing enzyme coated membrane on the ion selective gates of ion selective field effect transistor. These biosensors are extremely small, less than 0.1 millimeter square. 4. Amperometric biosensor. These electrode functions by production of current when potential is applied between two electrode, the magnitude of the current being proportional to the substrate concentration. The simplest amperometric biosensor uses the Clark oxygen electrode, which determines the reduction of oxygen present in the sample analyte solution. These are the first generation biosensor. These biosensors are used to measure the redox reactions. A typical enzyme being determination of glucose using the glucose oxidase. The principle of a mediated biosensor is shown. In this biosensor, a redox reaction is catalyzed by an enzyme is directly coupled to an electrode where enzyme is presented with the oxidizable substrate. The electrons are transferred from the substrate to the electrode via enzymes and redox mediator. In this biosensors, a oxidase replaces the oxygen replacement of the enzymes. The major problem of such biosensor is their dependence on the dissolved oxygen concentration in the analyte solution. This may be overcome by using the mediator, example ferrocene. These molecule transfers the electrons generated by the reactions directly to the electrode rather than reducing the oxygen dissolved in the analyte solution. These are called second generation biosensor. The present day electrode, however, removes the electrons directly from the reduced enzymes without the help of mediator and are coated with the electrically conducting organic salts. Enzyme electrodes. The enzyme electrodes are the new type of biosensor which have been designed for amphometric assay of potentiometric assay of substrates such as urea, amino acids, glucose, alcohol, lactic acid. The electrode consists of a given electrochemical sensor is in close contact with a thin permeable enzyme membrane capable of reacting with the given substrate. The enzyme is embedded in the membrane and produces oxygen, hydrogen H plus, NH4, ammonia plus, carbon dioxide CO2 or other small molecule depending on enzyme reactions. This is detected by the various sensors. The magnitude of the response determines the concentration of the substrate. 5. Optical bio typical enzyme based biosensors. Substance amines for meat freshness enzyme monoamine oxidase response time 4 minutes, range 50 to 200 micromole dm-3. Cholesterol oxidase response time 2 minutes, range 10 to the power 2 to 3 into 10 to the power 5 moles dm-3. Carbon monoxide CO acceptor response time 15 seconds, range is 0 to 65 micromoles dm-3. Glucose oxidase response time 20 seconds, range 2 into 10 to the power 3 to 3 into 10 to the power 6 mole dm-3. Penicillin enzyme penicillinase response time 25 seconds and range 1 to 10 micromoles dm-3. Sucrose enzyme invertase response time 6 minutes, range 10 to the power 2 to 2 into 10 to the power 3 mole dm-3. Uric acid enzyme uricase response time 30 minutes, range 5 into 10 to the power 3 to 5 into 10 to the power 5 moles dm-3. 5. Optical biosensors. These biosensors measures both catalytic and affinity reactions. They measure a change in fluorescence or in absorbance caused by the product generated by the catalytic reactions. Alternatively, they measures the changes induced in the intrinsic optical properties of a biosensor surface due to loading in it of dielectric molecules like proteins. In case of affinity reactions, a most promising biosensor involved luminescence used firefly enzymes luciferase for detection of bacteria in food or clinical samples. The bacteria are specifically lysed to release ATP, which is used for luciferase in the presence of oxygen to produce light, which is measured by the biosensors. 6. Bioaffinity sensors. The bioaffinity sensors are developed recently. It measures the concentration of the determinant, that is the substance based on equilibrium binding. This shows a high degree of sensitivity. These are of diverse nature because of use of radio label, enzyme label or fluorescent label substances. The principles of bioaffinity sensors is given. In this biosensor, a receptor is radio labeled and allowed to bind with the determinant analog immobilized on the surface of a transducer. When the concentration of the determinant are released, the labeled receptor forms an intimately bound complex with the determinant. Finally, the radio labeled receptor determinant complex is removed from the immobilized determinant analog, resulting in the release of concentration of labeled receptor. This is measured by the reduction in signal of labeled receptors. Gronow et al. In 1988 has discussed that the possibility of this type of biosensor or the use of lectin receptors for saccharide estimation, hormone receptor for hormones, drug receptors for drug, antibiotic receptors for antigen, and nucleic acid as a gene probe for inherited disease and fingerprinting. Acoustic wave biosensors. These are also called piezoelectric devices. Their surface is usually coated with antibodies which binds to the complementary antigens present in the sample solution. This leads to increased mass which reduces their vibratory frequencies. This changes is used to determine the amount of the antigen present in the sample solution. Eight electrochemical biosensors. This type of biosensors has been developed by using the electronic device such as a field transmitter or light emitting diode. The former measured charge accumulation on the surface and the latter photo response generated in a silica based chip as an alternative current. Hence the field effect transmitter measures a biochemical reactions at the surface and induced into current. Moreover, the field effect transmitters can be modified to ion sensitive or enzyme sensitive or antibody sensitive ones by using the sensitive ion enzymes or antibody respectively. 9. Whole cell biosensor, microbial biosensor. In this device, the entire immobilized whole cell of microorganisms or their organelles are used. These reacts with many substrates and show generally slow response. The immobilized Acetobacter vinylandi couples with the ammonium electrode shows sensitivity range between 10 to the power of minus 5 and 8 into 10 to the power of 10 mole dm minus 3. It measures the concentration of nitrate within 5 to 10 minutes minus 2. Application of a biosensor. In the beginning, biosensor was applied in the field of medicine and industry, but in recent years biosensors are becoming popular in many areas due to the small size, rapid and easy handling, low cost and greater sensitivity and selectivity. The application of biosensor in some of the area is described as below. 1. Use in medical and health. The biosensor have tremendous potential for its application in the field of medical science. In 1979, the first glucose analyzer used biomolecules of detection of blood glucose was commercialized by Yellow Springs Instruments Company USA. A device in a mini pump filled with insulin has been constructed to deliver insulin to diabetics based on glucose level of blood. 2. Use of pollution control. The biosensors are very helpful in environmental monitoring and pollution control since they can be mini authorized and automated. As far as quality control of drinking water is concerned, the monitoring biosensor are useful in monitoring of pesticide in water. 3. Use in industries. Generally, the spectrophotometer and auto analyzer are used to establish the substrate utilized and the product formed in the fermented broth. 4. Biosensor in military. The darker side of the biosensor application is to provide support to the military which with such a biosensor that can be detect toxic gases including the chemical warfare agents such as such biosensor have advantages over the traditional method of sensing of chemical.